Dr. Stefan Bohlander is joining us now at ASH 2013. He's from the University of Auckland in New Zealand. His research in the leukemia field took him from Germany to where he is now. And Doctor, thanks for being with us. Tell us about the significance of your research.
Well, any hematologist will know that diseases, especially acute myeloid leukemia, are a big problem. More than half the patients, in some instances 80% of the patients who are diagnosed with acute myeloid leukemia will eventually die. So there is a need first to understand this disease better, and then second to develop new treatment strategies and also develop new drugs for treating this disease.
You're faculty here at ASH for the Genomics 101 session. What can you share with us about this?
Well, the educational session is called Genomics 101 for the Practicing Physician. And of course the objective is to familiarize physicians with the new technologies that have evolved at an incredibly rapid pace over the last 8 years in genomics. So one of the striking developments is that we have really increased the information output that we get out of sequencing by an order of 7 to 8 orders of magnitude over the past 5 years. So this is a development that outstrips any development in the computer industry or any other industry that we have ever seen. So we're now able to basically do the work that took 13 years, just 10 years ago, to accomplish in 2 hours, sequencing a human genome.
Dr. Bohlander, it's got to be challenging for busy hematologists and oncologists to keep up with this rapidly changing technology.
Well, it's actually for the practicing physician, because time is limited, it's hard to keep up. And what we do that in ASH is trying to give a rough overview over these developments. Now, what we try in the type of more basic type of research, we try to understand the genetics of leukemia. The initial efforts are well known to any practicing hematologist. I mean, initially genetics was looking at chromosomes. So one of the protagonists in this field is Dr. Janet Rowley from the University of Chicago, who in 1973 discovered the 922 translocation in chronic myeloid leukemia, which leads to the production of the BCR-ABL fusion protein. And then in the year 2000, Brian Drucker was able to develop this inhibitor, which everyone knows, so it's imatinib, and that really has converted CML from a mostly fatal disease into a chronic disease. Some of the patients are on treatment for 13 years now. Now I'm more focused on acute myeloid leukemia, and here the genetics, also starting out with chromosomal analysis, proved to be much more complicated than in CML. So what we see is a multitude of chromosomal rearrangements, and now we've been able, using these next generation sequencing methods, really to look at individual tumor genomes, and we find quite a variety of different mutations that we know drive the disease. So what's coming out with all this is that the picture gets more complicated. We know more and more mutations, and what also makes it even more complicated, that every disease, every AML is sort of very individual. Just like we are all individuals, so every one of the diseases we treat is probably an individual. And in the past, we have treated AML mainly with one therapy, chemotherapy, and this has not changed very much since the 1970s, so for the past 40 years. But in the past 40 years, our knowledge about the genetics has, mushroomed and skyrocketed. So the next thing would, of course, be, and this is also in the development, to treat patients according to the genetic lesions they have in their disease. This is going to be a challenge, and it's sort of the problem I'm working on. So we are actively sequencing individual tumor exomes, so just the protein coding part, but also the genomes in the future. And then if we find the mutations, we quite often have the problem to say whether a given mutation is actually critical for the disease process or whether it's just what we call a passenger that was sort of, jumping on the bus when the disease got started. And to really tell these two things apart, the critical mutations, which we call drivers, and the passengers, we are planning, and we're actually doing, developing mouse models, and in Auckland also in the future we're going to develop zebrafish models, which are probably better suited for looking at drugs that will impact on the different models.
So your work is really based in personalized medicine.
Right, that's what we are hoping to do, to make the treatment more personalized. But as you know, there's a lot of hype about personalized medicine, but everything is more difficult when you get to it than you actually thought it would be.
What is the future of your research?
Well, I would hope to be able to write a manuscript about some of the models we have made, and maybe also have hints on how to influence these models by using different kinds of drugs, small molecule inhibitors. But basically just understanding how some of the leukemias work would be great. And what we see is that it's not enough to just take one mutation and then you have a leukemia. It is a cooperation of several mutations, 3 to 8 mutations that drive a leukemia. And so this is very challenging to model, of course, because an individual, say a mouse knock-in, it takes 2, 3, 4 years. And if you multiply this by 8, you can imagine how long it would take to model an individual leukemia. So we have to also find more efficient ways to do that.
Thanks for being with us, sir. Dr. Stefan Bohlander joining us here at ASH 2013 from the University of Auckland in New Zealand.